कोई भी नहीं आया मेरी लास्ट क्लास में क्योंकि चाह के भी नहीं आ सकते। हम लोग ये करेंगे। ठीक है, बहुत इजी है देखो। ऐसे चलो हम स्टार्ट करते हैं। हम हैं हम क्या All right, so we are going to start the last class. So today we will be dealing with some of the questions that we have dealt uh, throughout this course. We'll be revising some of the questions and we'll just recapitulate some of the important topics from each week's class. And that's all we are going to do today. It's just going to be 1 hour class today. Fine. तुम बोलना मत ऐसा। हाँ हाँ ऐसा ऐसा मत करना है। हम अनम्यूट कर रहे हैं। So I'll be presenting my screen. Okay. So in week one was just about the introduction. It was an introductory class where we have studied about the RNA world. So what is the meaning of RNA world? RNA world means learning and understanding all about RNA, what all RNA is about, and what are the characteristics of RNA, what are the properties of RNA, what are the functions of RNA. So, just a minute. So this phrase RNA world, it was first coined by Walter Gilbert in 1986, and RNA world means RNA world means a time when RNA used to perform both genetic and catalytic functions. So earlier, as the evolution has taken place, so in the earlier world, it was RNA only that served as a genetic material. Eventually, in the course of our evolution, DNA has evolved as a genetic material because DNA is more stable than RNA. RNA is not very stable. And that's why DNA has evolved as a genetic material in due course of time. But initially, RNA was the one which used to perform both genetic as well as catalytic function. So, the basic properties of RNA are that RNA can store genetic information. RNA can be a temporary copy of a genetic information. RNA can be a carrier molecule, RNA can be a working part of a cellular machinery, and RNA can be a catalyst. These are the basic properties of an RNA. So the first question, that is, name the RNA molecule which takes part in the formation of ribosome. mRNA, tRNA, rRNA, or guide RNA. What should be the answer? Which RNA molecule takes part in the formation of ribosome? Just a minute. Yeah. So, as we have studied in our last class, that it is rRNA, that is ribosomal RNA. So, mRNA is basically involved in the coding. So, when I talk about coding, what is the meaning here? Coding means that mRNA has the sequence that codes for protein. So, mRNA gives us the sequence. That is the role of mRNA. Fine. So, mRNA gives us the sequence. tRNA, it serves as an adapter molecule, and rRNA, it is involved in ribosome, takes part in ribosome, which is involved in peptide bond formation also, and in the formation of ribosome also it is involved. So it is rRNA. Next question is, RNA contains repeating units of deoxyribonucleotides, deoxyribonucleosides, or ribonucleosides. What should be the answer? So the answer should be ribonucleotides. Why? Because if you see RNA and DNA, the basic difference is that that the at two second position there is OH in the case of RNA. There is, just a minute. Okay. So I think today there is overlapping of the classes. So I got to know that the other TA is also taking class from 6 to 8 and mine was from 7 to 8. So there is an overlap. I tried to rectify with the committee, but I think at the last moment it cannot be done. So it doesn't matter. I've started the class because I have to start at 7 o'clock. So I've started the class, and uh, you're here. And if others who cannot join the class, it's fine. They can see the recording if they want to see. So what I am doing today is that in each week, what all we have studied, we'll be just dealing two three questions from each week. Okay, so I first started with week 1. So in week 1 we have studied about RNA world. Okay, so what is RNA world? So in earlier times, when uh, the genesis of the life form has taken place, at that time RNA has only served as a genetic material. So RNA world means that RNA used to perform both genetic as well as catalytic function. But in due course of evolution, in due course of time, DNA, it evolved as a genetic material, but earlier it was just RNA. And then we are just solving two questions, that RNA molecule which takes part in the formation of ribosome, it is ribosomal RNA, and RNA consists of repeating units of ribonucleotides. Next question is, at what rate does the RNA mutate as compared to DNA? Faster rate, slower rate, moderate rate, or depending on the medium? So at what rate it should mutate? It should always mutate at a faster rate. Why? Because RNA is unstable. DNA is more stable in comparison to RNA. RNA is less stable in comparison to RNA. And because RNA is less stable, RNA, it is less stable. Therefore, the chances of mutation or the probability of mutations in RNA are more. Therefore, RNA mutates at a faster rate in comparison to DNA. Then, yeah, true or false? RNA is able to act as better enzymes than protein. What it should be? It should be true or false? RNA is a better enzyme or a protein is a better enzyme? So it is false. Why it is false? So today, because just you are here, so I'm not asking questions, because then every question would be directed to you only. We'll try to be a little quick because we have only 1 hour and we have a lot to cover today. So why you think that RNA is not a better enzyme? Because proteins can contribute more variability. So if we have proteins as enzymes, because there are 21 amino acids, so many different kinds of protein combinations that can be formed, and therefore the variability in the enzyme function, in the enzyme structure, everything would be more. Therefore, proteins are better enzymes in comparison to RNA. And not only this, proteins are more stable than RNA. Therefore, this again counts to the fact that proteins are better enzymes than RNA. Then RNA cannot store genetic information. True or false? It is again false. So why it is false? Because RNA can store genetic information. In fact, in earlier times, RNA was the only genetic material of the body. But because of some obvious reasons that DNA is more stable, RNA has not evolved as a genetic material, but it can store genetic information. Okay. So next we comes to week 2. So in week 2 we have learned about that RNA can also function as enzymes. So in the first week we studied about the basics, the RNA world, what is the meaning of RNA world, how this term was coined, what are the different functions of RNA world in our RNA in our body. In week 2 we'll be dealing with that what are the function of RNA, what is the function of RNA as enzyme. So RNA can work as an enzyme. So what does it means? That it has a catalytic activity. RNA has a catalytic activity. And this catalytic activity helps RNA to function as an enzyme. For example, ribonuclease P, ribozymes, self-splicing introns. So these are all the examples where RNAs are function as an enzyme. Okay. So if I talk about ribozyme, what is a ribozyme? A, it's a ribonucleoprotein, a ribonucleic acid, nucleoprotein or peptide nucleic acid complex. What is a ribozyme? It's a ribonucleoprotein. That means ribozyme, as the name suggests, it is made up of RNA plus protein, and the protein part function as an enzyme. Okay. Then the peptide bond formed during protein synthesis in ribosome is by RNA, protein, tRNA or mRNA. So this we have studied in our last class also. We have studied in translation in depth the role of three uh, different RNA in protein synthesis. So I hope this question would be easy. So the peptide bond formation during protein synthesis is formed by protein, tRNA, mRNA or RNA. So it should be ribosomal RNA. Here it should be R. A would be the answer. Why? Because mRNA will serve the code which will be converted into amino acid. tRNA, tRNA will function as an adapter molecule which will bring the amino acid. And the actual peptide bond between the amino acids that would be formed because of ribosomal RNA. Okay. Next question is, okay, now we'll come to week 3. So in week 3, up till in week 1 and week 2, what we have studied? We have studied about what is RNA, then what are the different functions of RNA, how RNA can function as a ribozyme, how RNA can function as an enzyme, that all we have studied. Then we have studied in week 3 about the various post-transcriptional modifications that happen. So if I talk that post transcriptional modifications. So what does it mean? It means that after the transcription, when DNA is converted into RNA, that RNA that is formed is a primary transcript. That RNA which is formed is a primary transcript. What is the meaning of a primary transcript? That it is in a very naive form. It cannot, this primary transcript variant cannot code for protein in itself. So it needs to be matured enough so that it can be converted into a mature RNA, and that mature RNA would only serve as a coding sequence for the formation of proteins. So this primary transcript undergoes various kinds of modifications, or we can say this primary transcript is processed in a way that it gets converted into a mature mRNA. Okay. And what are those processing that takes place? Removal of the nucleotides, which means splicing. Addition of the nucleotides to the 5 prime or 3 prime end. At the 5 prime end, what we add the cap, methyl cap. At the 3 prime end, what we add a poly tail. Modification of certain nucleotides. So their modification means some nucleotides are getting acetylated, some nucleotides are getting methylated. So these are different modifications that takes place. So these all collectively called as RNA processing. So, and it is also very important to note that if, let's say, 50 primary transcripts are there, then not all 50 would be converted into mature RNA. Okay, very few RNA molecules are transcribed to final mature RNA. This is important. Okay, then RNPs we have studied just in week 3. We have also studied about RNPs, which are ribonucleoproteins. That means which is RNA and protein both, for example, ribosome. And in this week 3, we have also studied about tRNA processing. So if we'll just quickly look at the various RNA processing that takes place. So first is removal of nucleotides by both endonuclease and exonucleases. So if this is your primary transcript, some nucleotides are removed from the ends. Some nucleotides are removed in between the RNA. So the nucleotides that are removed from the ends, from the exterior, they are known as exonuclease. If EX for exterior, EX for exo. And the nucleotides which are removed from the inside, they are removed by endonucleases, which means inside. Okay. And this is a general process which is seen in both prokaryotes and eukaryotic RNA. So this is a removal of the nucleotides by endo. Endo means inside, enzymes that cuts inside the RNA, and exo means enzymes that are cutting outside the RNA. Then addition of nucleotides to 5 prime or 3 prime ends. That means this is, if this is your RNA at the 5 prime end, 7 methyl guanosine. 7 methyl guanosine. That means at the guanosine, the 7th position of guanosine, methyl group is added. So this is your capping. This is your capping. And at the 3 prime end, you have a tail of adenosine moieties. And because there are many adenosine moieties added at the 3 prime end, we call it as a poly A tail because there are many tails added. This is another kind of modification. What is the third modification? Modification of certain nucleotides on either the base or the sugar moiety. That means addition of nucleotides, removal of nucleotides, okay, and modification of the existing nucleotides also. Modification of the existing nucleotides, how it can be take place? It can take place by adding of some groups. For example, you are adding methyl group or acetyl group or something like this. So if a particular sequence is methylated, if I say a particular sequence is methylated, that means that sequence will not be expressed. Okay, no expression. So methylation is a mark that specifies that expression will not take place. Okay. So these are the 3 modifications that occurs after the transcript has been formed. And we have studied in detail about these transcripts. Detailed videos, if you have like if you want to revise any topic, you can go and you can see through the recorded lectures that are available on YouTube. Okay. So now the first question is, which of the following statement is not true regarding transcription or RNA synthesis in eukaryotes? Okay. RNA synthesis occur in the nucleus. Unlike DNA synthesis, the only selective sequence of DNA is transcribed to RNA. RNA synthesis requires a short stretch of RNA primers. DNA sequences, specific proteins, and small RNA regulate RNA synthesis. So, what should be the what should be the answer here? Question is, which of the following statement is not true? regarding transcription in eukaryotes. We are talking about eukaryotes. So eukaryotes means they have a well-defined nucleus and they have a cytoplasm. Fine. That means compartment of nucleus and cytoplasm is there in eukaryotes, but it is not there in prokaryotes. So in a prokaryotic cell, transcription, translation, replication, everything is happening inside the cytoplasm only, because there is no well defined nucleus. But in the case of eukaryotes, there is a well defined nucleus, and RNA synthesis occurs in the nucleus. And when a primary transcript is completely matured, when RNA processing is completely done, then that RNA comes out of the nucleus into the cytoplasm and translation occurs here. So translation in the case of eukaryotes, it occurs in the cytoplasm, whereas transcription in the case of eukaryotes, it occurs in the nucleus. So first statement is correct. Fine. Second statement is uh, yeah. Unlike DNA synthesis, the only selective sequence of DNA is transcribed to RNA. This is also right. Why? Because we have dealt this concept in detail when we were uh, doing this chapter. That in DNA, if this is your double-stranded DNA, then both the strands serve as a template for the synthesis of DNA. Both the strands serve as a template. But in the case of RNA, this is a double-stranded DNA. Only one of the strand of DNA serves as a template, not the other strand of the DNA serves as a template. That means, unlike DNA synthesis, only selective sequence of DNA is transcribed to RNA. This is correct. Okay. Then RNA synthesis requires a short stretch of RNA primers. Now, this is a wrong statement. Why? Because we know that RNA have catalytic activity. That means for DNA synthesis, we require primers. But for RNA synthesis, we do not require primers. And this is one of the advantage of RNA. That is why earlier in the ancient times, in the earlier times, RNA was the genetic material because there was no need of primers. But it has certain disadvantages also that is not of our concern right now. But this statement is wrong. Then DNA sequence, specific proteins, and small RNA regulate RNA synthesis. It is also correct. So C is the incorrect statement. Then RNA transcription occurs in 3' to 5' direction. What it should be? True or false? RNA transcription, it occurs in 3' to 5' direction or 5' to 3' direction? It occurs in anything. Always remember, make it a thumb rule. Whether it is replication, whether it is transcription, whether it is translation, everything is happening in 5' to 3' direction. Fine. So, this is false. Then, tRNA processing is done by RNase P, RNase H, RNase K, RNase A. What should be the answer? The answer is RNase P. A is the answer. Okay? Fine. Okay. Next question is, which of the following RNA serves as the regulatory functions, including splicing and gene silencing? Means, here we're talking, see, here we are specifically talking about the regulatory functions. Let's see the options. mRNA, tRNA, rRNA, or snRNA. So, which serves as a regulatory function here? Which of the RNA is serving as a regulatory function here? It is? Can you tell me? A, B, C, or D? What should be the answer? It should be? It should be D. Why? See, if you remember, even if you don't know the answer, you can try to negate the options. mRNA, we haven't studied anything about regulatory functions of mRNA, tRNA, rRNA. So itself, this is the odd one out option. So this can be the answer. One logic. Another logic is if you have even a little bit of idea about the non-coding RNAs. For example, microRNA, nuclear RNA, these non-coding RNAs are involved in gene silencing. And this small nuclear RNA is a non-coding RNA. Third way by which you can guess is splicing. So, small nuclear RNA are involved in splicing. Okay. So, small nuclear RNA are the ones that helps in the splicing of the introns. Splicing occurs in the nucleus, and these are the RNA that are also present in the nucleus. Ribosomal RNA, these are not present in the nucleus, they are present in the ribosome, and ribosome is present in the cytosol. Transfer RNA, they are not specifically present in the nucleus, they are taking the information and they are helping in translation. So again, their function is in the nucleus. Here we're talking about splicing. Splicing is an event which exclusively occurs in the nucleus. So from here also you can say that it is a nuclear RNA, so this would be the answer. So many logics, and every logic is concluding to one answer only, which is small nuclear RNA. Okay. Next question is, Rho factor is important for RNA transcription termination, recognition of DNA regulatory region, transcription elongation, and RNA transcription initiation. So, first of all, you should know that Rho factor is needed where? In replication, transcription, translation, where? It is required in transcription. Okay. Now, if you remember, if we talk about transcription, the termination of transcription happens in two ways. Transcription, one is row dependent and another one is row independent. So row dependent means row is a protein, it's an enzyme helicase which helps in the termination of transcription. And when there is no row protein, then what happens? Hairpin loop formation occurs, and this hairpin kinds of pulls the RNA away from the DNA. Okay, and this loop has after it has inverted repeats of AT rich sequence, because AT has two bonds which is easier to be pulled. So suppose if this is your DNA and if this is a kind of loop formation has occurred, so this loop will try to pull it down, and so this RNA would be detached from the DNA. So termination of transcription can happen by two methods, row dependent method and row independent methods. That means row factor is important for RNA transcription termination. The answer is A. Fine. Now next question. Sigma factor is unique for elongation of transcription, turning on specific genes, bacterial adaptation, or mRNA stability. What should be the answer? Elongation of transcription, sigma factor. Is it important for elongation of transcription? No. mRNA stability? No. Bacterial adaptation? No. Turning on specific genes? It might be the answer, although this is not the exact answer of this question. Sigma factor is recognition is important for what? This statement is not giving the exact answer, but out of all the statements, this is only the most nearer to the answer. How? See, sigma factor is responsible for recognition of the promoter sequence. Okay. It is responsible for the recognition of the promoter sequence. So in RNA polymerase, when we talk about a prokaryotic RNA polymerase. This is a prokaryotic. We have alpha, two alpha subunits. Okay. Then we have beta and beta dash. This makes the core enzyme. And along with this, when sigma factor comes, this becomes holoenzyme. And this holoenzyme actually goes to the promoter sequence. And the sigma factor of this holoenzyme, this sigma factor is responsible for the recognition of promoter region. And when this promoter region is recognized, then transcription happens. That means certain genes are switched on or turned on. That is why transcription is happening. So again, if we go to this question, we can say that turning on specific genes is the most correct statement out of the given statements. Okay. So B is the answer. Now we'll come to week 4. In week 4, we have studied about alternative RNA processing. Now, what is the meaning of alternative RNA processing? So, up till now, we have studied, matlab we are just revising. We have already studied all these things. So, if we talk about RNA processing, what we talk about? We talk about 5' capping. We talk about 3' end poly A tailing, or we talk about splicing, or we talk about some modifications like methylation, etc. These are the known ones, more famous ones. But these are the normal types of events or methods of RNA processing. But alternative RNA processing means that same pre-mRNA can be converted into more than one type of mature RNA. See, conversion of the pre-mRNA species into more than one type of mature RNA. How this can happen, we have already studied, but I'll just quickly recapitulate it. That if this is your pre-mRNA, okay, this is your pre-mRNA, and in this pre-mRNA, this is your intron 1, this is your intron 2, this is your intron 3, this is your intron 4, and this is your intron 5. So if these are your introns, that means this is your exon 1, exon 2, exon 3, exon 4. Let's say. And this is exon 5. Okay, we'll just consider this much. Now, when splicing is happening, splicing can happen in many ways. What is the meaning of many ways? Introns has to be removed. If this intron is removed, and this intron is removed, and this intron is removed, so what is there? We have E1 combined to E2 combined to E3. Like this we have. If this intron is not removed, I1 is not removed, we only have E2, only this intron is removed, and we have E2 which is combined with E3. If these two introns are removed, we have E3 combined with E4. That means you can see we can have different combinations of exons. And this different combination of exons ultimately gives us different types of matured RNA. Matured mRNA or RNA in general. This is one way of alternative splicing. What would be the other way of alternative splicing? Poly A tailing. So differential poly A tailing means we have already studied that in an mRNA, poly A tail is added when there is a poly A tail binding site. there has to be a poly A tail binding site, and after that poly A tail is added. Now in this mRNA, you can have a poly A binding site here, you can have a poly A binding site here. So if your poly A binding site, this poly A binding site is used, then the mRNA would be of a longer length. If this poly A binding site is used, then mRNA would be of a different length. So the use of different poly A binding site for the addition of poly A polymerase, polyadenosine leads to different length of RNA, which will create different types of matured RNA. And these are the two ways in which alternative splicing takes place. Okay, now quickly we'll see the questions. So, first question is, we have to be a little quick because a lot has to be covered. Okay. When pre-mRNA is spliced more than one way, it results in truncated RNA, incomplete RNA, RNA isoforms, or RNA concatemer. What it will lead to if pre-mRNA is spliced in more than one way? It will lead to different types of RNAs. That means different forms of RNA. That means RNA isomer. Fine. Now next question is, average size of an exon is, so the average size of an exon is 150 nucleotides. So important, remember it. Fine. Okay. Next question is, key reaction in RNA splicing is condensation, hydrogen bonding, trans-esterification reaction, or coupling. So if you remember, we have talked about that in RNA splicing occurs to two sequential trans-esterification reaction. So if this is your intron and this is your exon E1, exon E2. This is the intron exon junction. This is an intron exon junction. This junction we call it as a 5' and 3'. Why? Because this is the direction of the whole mRNA. This and this junction. And in between somewhere we have a branch point. Here we have AU, here we have GC. So first this branch point will attack here. In branch point we have an A. And there is a lariat formation, and then this formed lariat will attack at the 3' end, and in a complete lariat form, your intron is removed. So this attack and this removal at the 5' splice site, here you have first trans-esterification reaction, and when this attacks at the 3' end, you have a second trans-esterification reaction. So there are two trans-esterification reaction in RNA splicing between the two branch point, 5' branch point, 3' branch point. So in an intron, you have splice sites. So you have a 5' splice site, you have a 3' splice site, and in between you have a branch point which has an A. Okay. Janela, you want to say anything? Fine. Now, which best describes alternative splicing? Using multiple proteins to create one gene? Using one gene to make multiple proteins? Using multiple proteins to make multiple genes? Using one mRNA to make multiple proteins? Can you answer this question? Think. B. Okay. Using one gene to make multiple proteins. No. Why? I understood your concept. Your concept is correct, but what you have to pay attention is fine. See, when we talk about splicing, we are talking about the cutting of RNA into different in a way that it makes different kinds of proteins. Fine. So we are talking about RNA. We are not talking about gene. Gene means which has introns and exons both. Okay, in your basics, if you remember, gene has introns also and gene has exons also. But in alternative splicing, we do not talk about genes. We talk about RNA. Specifically, we're talking about mRNA. Let's say over here. So the answer would be D. That is using one mRNA to make multiple proteins, not using one gene. Gene is a DNA which has introns, exon, 5' UTR, 3' UTR, many things are there. Here we're talking about RNA because RNA is spliced. It's a post-transcriptional event. Alternative splicing is a post-transcriptional event. Okay, not a post-replication event. Fine, I think it is clear to you. Okay. So now we come to week 5. In week 5, we have studied about the importance of introns. So we know that there is a high cost which is required for the maintenance of introns, but still our body has introns because introns allow the expressivity of the gene. Introns dilute the genes, especially the ones which are prone to mutation, and pre-mRNA can be spliced in more than one way, which can produce isoform, which is only possible because of introns. Introns increase transcriptional outputs and translation outputs as well. And why we need to have introns? Because introns helps in alternative splicing. It helps in promoting transcription. It helps in increasing the accuracy, that means the fidelity, and exons shuffling, that means exon shuffling means just alternative splicing only, increases recombination to create more products. So these are all, we have dealt all these examples in detail. I'm not repeating it right now. You can go and see the recordings if you have any problem. Just revising that in week 5 we have dealt more focus on why there is a need of introns and why introns are important. Okay, so the RNA component deleted from the pre-mRNA after trans-splicing is called as lariat, polypyrimidine, SL sequence, or outron. Anybody remember the answer? Fine. So we have dealt with two kinds of splicing. One is trans and other one is cis splicing. Now, what is the difference between trans splicing and cis splicing? That means in cis splicing, whatever splicing machinery and whatever splicing is taking place, it is within the same mRNA. But trans means it is attacking a different mRNA. It is a different mRNA. And in cis, the portion that is deleted, it is known as a lariat, and in trans it is known as an outron. So what is trans splicing, what is stress? Again, we have given a very good amount of time to it. Just remember, same mRNA, cis, two different mRNAs, if it is involved, it is known as trans. So the answer is outron. Next question is, what is the main function of tRNA? Proofreading. It inhibits protein synthesis. It identifies amino acids and transport them to ribosomes. None of the mentioned. So what is the function of tRNA? It, we have just studied in the last class, it identify the amino acids and transport them to ribosome. That means it acts as an adapter molecule. Fine. Okay. Next question is, the 3' end of an intron is marked by donor site, acceptor site, AT rich site, or branch site. So 5' end, 3' end, branch point. So 3' end would be, what should be the answer of this question, you tell me? Anybody who want to try? 3' end, what is the middle point? Can you tell me what is the middle point known as?
Branch point, sir.
Branch point, yes. And 5' end? Can anybody tell me? Even I am getting confused. That's why I'm asking you guys. Okay, we'll just leave this question because even I am getting confused here. We'll first solve all the questions and then we'll come to this question back. Okay. Okay. During splicing, which of the following site is not recognized by spliceosome? Donor, branch point, acceptor, G-rich region. So we have already studied that one site is known as a donor site, other site is known as an acceptor site, and in between we have a branch point. So donor site, acceptor site, and your branch point, each and every part is involved in splicing and each and every part is recognized by the spliceosome. But no way we mention about the G-rich site. So G-rich site is not recognized by the spliceosome. Okay. In week 6, we have studied about the small different kinds of nuclear RNA such as snRNA, rRNA, miRNA, saRNA, their processing, their export, and their functions. So here you can see we have studied about how introns can be gained. It can be gained by transposition of the introns or the insertion of the introns with the help of transposomes, which are also known as jumping genes. Okay, and we have studied about the different pathologies which results from the splicing defects. So here what is important? Your spinal muscular atrophy, this is important. Pigmentosa, this is important. Beta thalassemia, this is important. Okay, so whatever I am marking, these are important ones. Fine. Okay. Then we have also studied about the difference between the nuclear import and nuclear export and the involvement of RAN GTP, RAD GTP, etc. and the cytoplasmic transport, how it takes place, the various mechanism we have studied about this. So this is all we have studied, just to recapitulate what all we have studied in these classes. So FG nucleoproteins are located in nuclear membrane, nucleolus, nuclear pore complex, or cytoplasm. Where they are located? You can see here in this photo. This is a nuclear. See here. This is a nuclear pore. Okay. And cargo means whatever protein it is there. And this cargo is recognized by the FG repeat domain, or this is, yeah, the FG repeat domain. And then it gets inside. You can see here the transport receptor, first it binds with the cargo, and when it binds with the cargo, the FG repeat domain, it recognizes it, it keeps it inside, and then it is transported back. That means this FG repeat domain, it is present where? It is present in the nuclear pore complex. So this FG nucleoproteins, it is present in nuclear pore complex. Then a disease that is the result of defective mRNA export. This is important question. Which disease is the result of defective mRNA transport? Osteogenesis imperfecta. Remember this. Okay, we have already dealt this question, just to revise you. Then, nucleoporins are not important in RNA transport. Are nucleoporins important or not? They are very important because they are only actually the ones which are helping. Yes, fine. False. I'm sorry, I'm not able to see the answers, but it's very good that you are attempting and trying to answer because we have less time today and we have a lot to cover. That's why I'm trying to address the questions a little fast. So the answer is false. Sorry, true. Huh. The answer is false because nucleoporins are very important in RNA transport because they are the ones which are actually helping in the RNA transport and recognition. For example, the FG repeat sequence. The FG repeat sequence is a nucleoporine which is present in the nucleopore complex, which is important for transport. Then we came to the mechanisms of mRNA decay. So there are three different mechanisms by which mRNA decay can take place. What are those mechanisms? De-adenylation dependent mRNA decay, deadenylation independent mRNA decay, and endoribonuclease mediated. That means first there is deadenylation that takes place and then mRNA is decayed. Here no deadenylation takes place and this is the majority that takes place. Okay. The question is the introns are degraded due to specific enzymatic sequence, unprotected ends, lariat formation, or endonuclease activity. Why introns are degraded? Introns are degraded due to unprotected ends. Always remember, this is an intron, or not only just intron, you just say it as any RNA or mRNA. So there has to be some kind of protection. So in mRNA, we have a protection in the form of capping and tailing. If capping and tailing are not there, then they can be easily attacked by exonuclease. So therefore, if they have unprotected ends, they are very much prone to degradation. So the answer is B. Second, which of the following is not a step to degrade mRNA? Deadenylation, 5' to 3' exonuclease activity, 3' to 5' exonuclease activity, or site specific degradation. So which is not an event? You can see here endoribonuclease mediated decay. It also takes place. Deadenylation dependent takes place. Here you can have all 5' to 3' exonuclease and 3' to 5' exonuclease and deadenylation independent events. So from here we can say that site specific degradation is a little difficult. Why? See, this site specific degradation in exceptions this can also happen. But here if you look the combinations which is given, deadenylation, it takes place. 5' to 3' exonuclease, if ends are unprotected, it can take place. 3' to 5' exonuclease activity, if ends are unprotected, it can take place. At a particular site, degrading is a little difficult because then endonuclease have to work. And in general, the efficiency of exonuclease is a little more than endonuclease because exonuclease can right from the ends, they can start their activity. Okay, so entering the open gate, entering a home through a open gate is easier than entering a home by cracking a wall. So just remember that endonuclease have to crack a wall to enter the gate, and exonuclease can just enter through open doors. And open door means unprotected ends. Fine. Then we studied about three kinds of decay: nonsense mediated decay, non-stop decay, and no-go decay. So in nonsense mediated decay, what happens? That a stop codon comes, and because of the stop codon, the decay happens, and the resulting mRNA is of a shorter length, which is not a good mRNA, which is degraded. Non-stop decay. What is the meaning of non-stop decay? Here you can see it can happen in two ways. First, the decay can happen either from the 5' end or the decay can happen at the 3' end. Okay. So if 5' end decay is there, then always remember, I'm just marking here for your reference. Ski 1 is important, and when ski 1 is absent, then it happens from the 3' end. Sorry, if ski 1 is there, yeah, I'm correct only. So ski 1 is there, so it would be 3' to 5' decay. Okay, I said the opposite. If ski 1 is there, then 3' to 5'. If ski 1 is absent, 5' to 3'. But in both the cases, what is important is the activity of exosomes. Always remember that exosomes will start the decay from the 3' end, and XRN1 will start the decay from the 5' end. So this exosome is at the 3' end and XM1 is at the 5' end. Then no-go decay. No-go decay means what? That ribosome is halted here. You can see there is stalled translation. Non-stop decay means that there is no stop codon. Here there's stop codon is there. Therefore, it starts the decay. Here, no stop codon is there. So it just keeps on translating, translating, and translating. But it cannot translate that much. So that is your non-stop decay. No-go decay means that ribosome cannot move, ribosome cannot go. So then comes your no-go decay. Exosomes always uh, work at 3' end, and XRN1 always works at 5' end. Okay. Okay. Now these are already marked, but that's fine. So nonsense mediated RNA decay occurs when, so when nonsense mediated RNA decay occur, when RNA has a premature stop codon. Okay. If it has a premature nonsense, so nonsense mutation means if any codon is replaced by stop codon, then that is a nonsense mutation. Likewise, nonsense decay means RNA has a premature stop codon. Then non-stop decay means protein synthesis do not stop at all. So nonsense protein synthesis stops at a premature time. Non-stop protein synthesis is not stopping at all, and no-go ribosome is stalled at the point. Fine. Then we have studied about dosage compensation and X chromosome inactivation. So dosage compensation and X chromosome inactivation means that if X chromosome is there in females, one of the X chromosome is inactivated to compensate the expression of the genes. That is known as dosage compensation. Okay. Now quickly try to answer this question with what is the class of zist RNA? Zist RNA is a NNC RNA, non-coding RNA, long non-coding RNA, shRNA or piRNA. This is an important question. Always remember, Zist is a long non-coding RNA. Fine. Then, bar body is the active X chromosome, inactive X chromosome, active Y chromosome or inactive Y chromosome. So always remember, there is no such thing like inactive Y chromosome. Why? Because Y is only one. Y is only present in males. That too just one. So there is no need of Y to be inactive because X is present twice in females. That is why X needs to be inactive. So bar body is the inactive X chromosome. Okay. Then there are different mechanisms of dosage compensation in different species. In mammals, you have X chromosome inactivation. One of the X is inactivated. In C. elegans, both the X are repressed. In Drosophila, this X is hyper expressed so that it compensates for these two. Fine. These are the different mechanisms of dosage compensation. And then we studied an example of X chromosome inactivation in calico cat. Okay. So calico cat are almost always females. This is very important. I'm just pointing the important points from all the topics. Fine. Dosage compensation helps to balance gene expression. Is it true or false? It is true. Dosage compensation always helps to balance gene expression because if one of the X chromosome is inactivated, so in males also expression is coming from one X chromosome. In females also expression is coming from one X chromosome because one of the X chromosome is inactivated. So it always balances the gene expression. What is the class of zist RNA? We already studied LNC RNA. Okay, these questions are repeated. Bar body is the inactive X chromosome. Fine. Okay. The best genetic mosaicism is observed in which of the following? Tobacco, fruit fly, dragonfly, or cat? What should be the answer? The answer should be
Cat.
cat. Why? Because we have studied here. Calico cat is the example. You see, it is a genetic mosaicism. Why it is there? Because X chromosome inactivation is random. Always remember, X chromosome inactivation is random. So what happens? There is random inactivation of X chromosome in different cells of blastula. In blastula, it is not that same cells are getting inactivated. It is random. So some cells are getting inactivated, some are not getting. Therefore, you get this mosaic kind of pattern. Fine. Then in week 10, we have studied about that this is important actually. How's RNA interference versus miRNA translation? So both are involved in silencing, but siRNA siRNA causes the cleavage of the target. siRNA, it causes cleavage of the target, whereas miRNA, it just causes the repression. Why? Because perfect base pairing happens in the case of siRNA. So in the case of siRNA, there is perfect base pairing, whereas in the case of miRNA, incomplete base pairing happens. Therefore, miRNA cannot cleave the target, but it will cause the translational repression. So now, what is hot air? Hot air is a non-coding RNA. If you remember, we have studied in detail about hot air. Genomic imprinting is seen in all living organisms. No, genomic imprinting is not seen in all living organisms because imprinting of genes occurs. There are specially genes here where imprinting occurs. What is the meaning of imprinting? That if a gene codes for two alleles, then allele of one of the parent is permanently repressed. It is already known that this particular gene would be inherited from one particular parent, either mother or father. So that is your imprinting, that the expression of that particular gene is imprinted to occur from the allele of mother or father. That is imprinting. But it is not seen in all the organisms, otherwise there would have been no variations. So because in nature variations are there, that is why we can say that genomic imprinting is not seen in all the organism. It is false. Then RNA interference mediated gene silencing is an ATP dependent procedure. True, it is an ATP dependent procedure. Then in week 11, what we have studied? In week 11, we have studied about telomeres. So what are the telomeres? Telomeres are basically the repeat sequences which are there to protect the ends of the chromosomes. Okay, telomere protects the ends of the chromosomes, and basically remember this point, telomeres are added because of the enzyme telomerase, and telomerase is an enzyme which is capable of renewing or adding the telomeric repeat to the chromosome, and these telomerase are nothing but a reverse transcriptase. This is important. Second is, what is the repeat sequence? TTAGGG. Remember this. I'm just highlighting the important points that I think are important. Okay. Then, question is, identify the correct telomeric repeat sequence. We just see here TTAGGG. That means TTA, यहाँ पे ये G होगा. TTAGGG. So, if there it is G, this is the correct answer. Then, telomere is a repetitive sequence present anywhere on the DNA. What it should be, true or false? It is false. Why? Because telomeres are always present at the end of the chromosome and not anywhere in the chromosome. Fine. Telomere length is maintained constant in all chromosomes. True or false? Again, false. Why it is false? Why it is false? Because telomere length is not maintained constant. It is maintained constant in germ cells, in some of the stem cells, and in cancerous cells. Okay. Otherwise, in somatic cells, what happens? In somatic cells, with each round of cell division, the length of the telomere gets shortened and shortened and shortened. That means telomere length is not maintained constant in all chromosomes. It is maintained constant in germ cells, in cancerous cells, and in some of the stem cells. Okay. Then we have studied about epitranscriptome and protein synthesis. Now, epitranscriptome means it includes all the modifications that happens in the RNA. So, the sum total of all the modifications that happens in the RNA, it is known as epitranscriptomes. And here we have studied one of the very important modification of epitranscriptome, that is N6-methyl adenosine. And this modification can be occurred or can happen because of the three kinds of sequences: reader, writer, and eraser. We have studied in detail about reader, writer, and erasers. So readers are the factors that recognize, okay, writer, reader, and eraser. We've studied like this. Writer, they recognize the they matlab they do the modification, just like writer write it. So writer, they do the modification, that means they're adding the methyl group. Reader, they recognize the modification, and eraser, it erases the modification. Just like a writer writes a poem, somebody reads it and somebody erases it. Similarly, a writer is a protein that adds the methyl group, that means it is writing the modification. Reader, it reads the modification, that means it is recognizing the modification. And eraser, it erases the modification. Fine. So that is your reader, writer, and eraser. And then, which of the following is a frequent RNA modification? Deadenylation, ubiquitination, N6 methyl adenylation, or simylation? What is the answer? You can see here, N6 methyl adenosine is added. That means N6 methyl adenylation is a frequent RNA modification that occurs. Then, factors that cause an RNA modification is called as, that cause the RNA modification. That would be writers, because they're writing the RNA modification, therefore they cause the RNA modification. So with this we have completed. So just one query that we had, if anybody knows the answer, they can tell me. uh, just tell me, if this is your intron, this is your branch point, this is the 5 prime splice site, and this is the 3 prime splice site, which arm is known as a donor arm and which arm is known as an acceptor arm?
3 prime is the acceptor.
3 prime is the acceptor. That is what I'm also thinking. I'm just confirming somewhere. Yes. So 3 prime site is the acceptor arm. This is your donor site. This is your acceptor site. And branch point which has an adenosine. This has GU. It has AG during splicing. So with this, we have completed the course. I understand that you have 2 classes today and the timings were overlapping. So students who even wanted to join, they could not join, but don't get disheartened. You will have your recorded lecture. You can go through the lecture and you can see. And I'm just writing my email ID here just in case if you have any query. This is my email ID. So in any query related to questions that we have solved, or in general, if you have any question regarding uh, what you have to choose for your higher studies, where to go, what not to choose, or any kind of competitive examination, or anything related to life science or research, you are always free to ask me. You can always, ma'am, can you explain about the exam pattern? Okay. So what I think is that you will have, just see whatever questions that you are doing in your assignments and whatever question that I am teaching you, same kinds, the same would be the exam pattern for your examination also. Up till now it has been the same. If sir does something new this time, that I can't tell you, but up till now it has been the same. So do not worry. If you have studied well, then see, there are in this course, always see, there are so many detailed information which is given, but you need not to go to all the detailed information for your exam. For your knowledge, it is important. For your exam, I am telling you why. You can go through the questions of each topic. Okay, whatever assignment questions that you have dealt with, whatever questions that we have discussed in the class about a particular topic, that can tell you how depth you have to go to a particular topic. Okay, so suppose if so many XYZ names are given, but you see the questions, are they asking such questions? No. Then just you can avoid it for your exam per se if you don't have time. Otherwise, every knowledge is important, everything is important. Okay, so with this, we'll end the class. It was just a 1 hour class. So it's been, I think it's been already, I don't know, it's been more than 1 hour. I wanted to take a 2 hour class, but I also have some work, so I'll go there now. So all the best for your exams. Hope you all will do well. It was very good teaching and interacting with you guys. We all learned a lot through this session. And especially thanks to the students who have always stick to the class from the first till last. It's really appreciable. And all the best for your future and all the best for it. Chalo, bye then. Janela, you can leave. I think there is some problem here. So something has hanged out, so I cannot leave the class.